Okay, here is the same figure that is in your extra credit. This is a picture of the light-dependent reactions of photosynthesis. So the light energy comes down, hits photosystem 2, which excites an electron. It goes whee, and then it falls through this electron transfer chain, and that pulls more hydrogen into the thylakoid compartment. Then the electron is transferred to photosystem 2, light energy comes down, and it goes whee again, falls through another electron transfer chain, and it makes NADPH, which you need later. Now this guy is losing electrons to photosystem 1, so it has to pull electrons from water. This makes water fall apart into hydrogen and oxygen. The oxygen gets given off as a waste, and the hydrogen stays inside the thylakoid. Now you're building up a pretty powerful concentration gradient of hydrogen. There's more hydrogen in here than out here. So the hydrogen really wants to leave the cell. The only way it can do it is through this passive transport protein called ATP synthase. So the hydrogen will come out of here, and as it comes out, each hydrogen allows this enzyme to do a reaction, and it makes ATP. So for every hydrogen ion that goes through, you get ATP. Now the NADPH that we made at the after photosystem 1 is used to make sugars. When the cell has too much NADPH, or it doesn't need to make more sugar, photosystem 2 stops giving its electron to photosystem 1. So photosystem 2 will take its electron back and keep making ATP. So here is a picture of regular non-cyclic photophosphorylation, what I showed you before, where photosystem 2 gets electrons from water, they get excited, electron transport chain, excited again, electron transport chain, NADPH. Cyclic photophosphorylation, it just keeps going back and forth to photosystem 1. Now we're still creating a hydrogen ion gradient, so we're still making ATP, but we're not making any of the NADPH we need for the sugars. Now that we've covered the light-dependent reactions, we are on to the light-independent reactions. These are the ones that we take the ATP and the NADPH that we made in the light reactions, and we use these products to make sugar. The main enzyme involved in this process is called rubisco. So the light-independent reactions, or dark reactions, do not use light to excite electrons. This is also named after Kelvin and Benson, the co-discoverers. And these reactions, or series of reactions, take carbon atoms from carbon dioxide and stick it onto a 3-carbon sugar to make glucose. This is a process called carbon fixation. So here's the Kelvin-Benson cycle in cartoon form. If you ever take biochemistry, you will have the esteemed honor of learning all the steps, but for this purpose, don't worry about it. So the carbon dioxide comes in here. There's a series of chemical reactions that requires NADPH, and it requires ATP, and it makes glucose. Now, there are 3 different types of plants out there, and they all do the Kelvin-Benson cycle in different ways. C3 plants can only use the Kelvin-Benson cycle to fix carbon. So the only way they can make glucose is by doing that cycle of reactions I showed you on the last slide. These plants have mechanisms to keep from drying out, like a waxy cuticle on their leaves and pores that close to prevent the leaves from losing water. Because if a plant dries out, it's toast. However, when a plant closes its pores to prevent the carbon dioxide, it prevents the carbon dioxide from entering the cell and the oxygen from leaving. So the oxygen starts building up inside the cell. When the oxygen concentrations get too high, the enzyme rubisco gets confused. Instead of adding carbon dioxide to this 3-carbon sugar at the start of the Kelvin-Benson cycle, it adds oxygen. So instead of making glucose, it makes junk that has to get broken down by the cell. And this is, at the end of this process, carbon dioxide is a byproduct that's given off. So the plant is losing carbon instead of fixing it. So if you're a C3 plant and you're living in Coos Bay, Oregon, you're probably going to do just fine. It's cool enough and moist enough here that you won't have to close your pores for too long. But what about if you live in a drier area? C4 plants okay, so C3 plants, what we've been talking about before, aren't very good at doing photosynthesis on dry days. So when it's dry and they have to close their pores, they mess up. And this process where they're adding oxygen to the sugar, called photorespiration, can limit the plant's growth, especially in dry areas. So up in Bend, you wouldn't see a lot of C3 plants. You'd see C4 plants. C4 plants are called C4 because they use a 4-carbon sugar to start the Calvin-Benson cycle. So they use a different sugar. In the mesophyll cells, so in part of the leaf, the 4-carbon sugar is fixed using an enzyme other than rubisco that does not malfunction in the presence of a lot of oxygen. The C4 cycle also uses rubisco, but it keeps the rubisco in another cell away from the oxygen. So it's like it keeps it in another room. So an example of a C4 plant is corn. You can grow corn in drier areas, but not too dry. You wouldn't see corn in a desert. What you would see in the desert is a cam plant. Succulents like jade plants and sedums and that sort of thing, and other plants that live in very dry, hot areas have an alternative way to fix carbon. This pathway is called the crassulacean acid metabolism, or CAM, C-A-M. They use the C4 pathway, like in the C4 plants like corn, plus the Calvin-Benson cycle. But instead of doing it in different parts of the cell, they do it at different times of the day. C4 plants will open their pores in the cooler night and fix carbon on the C4 pathway. The 4-carbon sugar is stored in the plant's central vacuole, and the next day the 4-carbon sugar goes out of the vacuole and is broken down into carbon dioxide. The carbon dioxide then enters the Calvin-Benson cycle. So it solves the problem by doing it at different times of the day. So this one shows C3 plants. These are the plants where on dry days they wind up doing the photorespiration. Here's how C4 plants do it. They separate the carbon fixation from the Calvin-Benson cycle by doing it in different cells. And the CAM plants do it at different times of day. They fix the carbon at night and do the Calvin-Benson cycle during the day. I believe that's it for photosynthesis. Have a great day.